Well, the study was to see what kind of prognostic impact minimal residual disease detection would have in infants with ALL and then KMT2A-rearranged ALL, because we know that in older children it's has a very strong prognostic value, but in infants it wasn't studied for so much. So that was the main goal, to see if there was also prognostic value in these babies. What we did, so in the infant 06 protocol, MRD was measured several time points. Only not all patients that were included, we had MRD available, but of more than half of them. And MRD was measured with qPCR, and the main target for qPCR was the genomic fusion breakpoint sequence. In this study, we saw that MRD is also in these KMT2A-rearranged infants, MRD is very prognostic for survival, and especially MRD at the end of induction. But we saw, because the infant 06 was a randomized trial between a lymphoid consolidation and a myeloid consolidation. And what we saw was that MRD was very prognostic in the patients that received a lymphoid consolidation, but it was not prognostic at all in patients with a myeloid style, getting a myeloid style consolidation. So actually that meant that patients that had high MRD at the end of induction had a very poor outcome when treated with lymphoid style consolidation. So, and we saw that those patients with high MRD at the end of induction had better survivals when treated with the myeloid style consolidation. And the other way around, patients with negative MRD had very good survival when treated with the lymphoid consolidation, and a worse survival when treated with the myeloid style consolidation. So actually what this says is that at the end of induction, MRD can select those patients that will benefit from a more lymphoid style consolidation and patients that will benefit from a myeloid style consolidation. So actually that is one of the main results that came out of this study. And furthermore, we also saw that if patients are still MRD positive at the end of consolidation, this was also prognostic for survival, and it was not impacted by the consolidation treatment given. So MRD is minimal residual disease, so it's very sensitive detection of residual disease. And while using qPCR, one can detect 1 in 10,000 to 1 in 100,000 cells. And in this study, the main target was the genomic fusion breakpoint sequence. So that's a very patient-specific target. And also due to that, there is no background expression of this marker or no false positivity. So this means that with this technique, you can really sensitively measure how much residual disease is there. And what we see that if residual disease is still detected after consolidation, for example, this is prognostics for survival, as in older children. We will use indeed these results for the next protocol, and so in that protocol, patients that react good on the induction therapy will receive a lymphoid style consolidation, and patients that still have high MRD at the end of induction will receive a myeloid style consolidation. And because we know that patients that are still MRD positive at the end of consolidation, we will also implement a kind of experimental window for those patients, because the survival is so bad for these patients that we want them to be eligible for experimental therapy. So that's how we are going to incorporate MRD in the next protocol.